Good morning student. In this video we are going to discuss about the voltage gated sodium channel, its structure, activity and selectivity. So what are ion channels? Ion channels are pore forming membrane proteins. They are the integral membrane proteins and they are formed by assemblies of several proteins or subunits. So such multi subunit assembles usually make a circular arrangement of identical or homologous proteins closely packed around a water filled pore through the plane of the lipid bilayer membrane. So from its name it is clear that it should be a channel, and the pore of the channel is formed by the arrangement of some proteins, subunits of a protein, and they will allow the passing of ions. So that's why they are known as the ion channels. Now there are two types of ion channel. One is a gated channel and not gated channel. The gated means the pore is whether any gate is present in the pore or not. If the gate is present, then they are known as a gated channel. And opening of closure of this gate, it depends on either on the change of voltage or it depends upon a ligand or chemical. If it depends on voltage change, then it is known as a voltage gated ion channel. If it depends upon the binding of some ligand or chemical, it is known as ligand gated ion channel. Here in this video, we are going to discuss about the voltage gated type of sodium channel. Sodium channel can have leaky channels which don't have any gate. But here especially we are discussing about the voltage gated sodium channel. Now here we can see the picture of some ion channels. First picture is showing it is of the potassium channel. It is formed by the tetramers. That is it is a tetramer. It is formed by 4 subunits and pore is in the central part. Second channel is the ligand gated acetylcholine receptor channel. Here it is formed by the it is a pentamer and the pore is in the central part. Third C is the chloride channel from the CLC family, and it has intracellular pore in each subunit. Fourth channel is the aquaporine water channel. It is tetramer and with intracellular channels in each of these subunit. Now look into the structure of sodium channel. So if we before we look into the structure, we must know few points. The voltage gated sodium channel usually exists in three forms. One is a closed form, open form and inactivated form. Closed form when its activation gate is closed. When due to depolarization or change of intracellular negativity to more positivity, there will be voltage change and it will cause the opening of the activation gate and the channel will be opened. Open means the sodium ion can pass through it freely. So in the first picture you can see this activation gate is closed. So due to depolarization or intracellular positivity, so there will be opening of voltage gated activation gate. So this opening is voltage dependent. After that there will be time dependent closure of the inactivation gate. In this picture we can see another gate is there. It will close the pore. So although the activation gate is open, there is no more sodium will enter into the through this channel. So that state of channel is known as inactivated state. After inactivated state, it will again move to the activation. Closure of activation gate will be there and it will move to the closed state. So it will have three states: closed state, then there will be voltage dependent opening of uh, activation gate, so it will become open. Then there will be time dependent closure of the inactivation gate, then it will be closed and again it will go back to the closed state from inactivated, and then again you will go to the closed state where the activation gate is closed. Now we want to know in which part of these subunits what is the which one is the activation gate, which one is the inactivation gate. So this is a structure of voltage gated sodium channel. It has got two subunits, alpha and beta subunit. Alpha subunit has 4 transmembrane domains. You can see 1, 2, 3, 4. They are actually surrounding the pore. Now each of these transmembrane domain they contain 6 transmembrane units. You can see here from S1, S2, S3, S4, S5, S6. So they have 6 transmembrane units. Now among them S4, the blue one, you can see that it had loss of positive charges. it will act as a voltage sensor. It will actually say the activation gate, now it is time to open, or it is time to close. So it is actually the sensor. Between the S5 and S6, the actually the pore is present, and there is a loop of protein, P loop, which acts as the selectivity filter. What is selectivity filter, we will say later. Now between the S6 of domain 3 and the S1 of domain 4, there is an hydrophobic amino acid which is known as IFM moiety. Now what is the function of this IFM moiety, we will see later. So this is the alpha subunits. In another picture, where alpha subunits, you can see that they are organized in 4 homologous domains, 1 to 4, each with 6 transmembrane segments, S1 to S6, 24 transmembrane segments in total. And the pore forming segments are formed by S5 and S6. So here we can see the details of the sensor, that is S4, and also the IFM moiety which is present between the S6 of domain 3 and S1 of domain 4. Now what happens when the channel is closed? You can see that at the S4 there are 2 hydrophobic amino acids which are indicated by red color. Two small round things. They are the hydrophobic amino acids alanine aspargine. When the channel is closed, when the activation gate is closed, they are covered, not exposed. Okay. Now this IFMT or IFM moiety, it has got an attraction for this hydrophobic amino acids. So when due to intracellular positivity, somehow through the leaky channels, when the sodium ions are entering inside the cell, and the intracellular negativity, it is changing, so to depolarized state, more positivity inside, so that will be sensed by S4. We know that it has lots of positive charge, so S4 will be pushed up. Okay, so there this, when membrane inside becomes positive, it pushes the positively charged containing S4 upward or outside, and this actually cause the activation gate to open, and so this is the open state and sodium will enter. When the activation gate is open, that means these alanine and aspargine or hydrophobic domains, they are exposed. So IFM moiety attract this hydrophobic domain. So there, after certain time, there will be time dependent bond formation between the IFM and this alanine aspargine. So this is actually what happens due to that is the closure of the inactivation gate. Now first it was closed, and these alanine aspargine were unexposed. Then due to the upward movement of the sensor, there will be opening of activation gate, that is the open state, and after certain time, due to the formation of bond between IFM and the alanine aspargine, the inactivation gate is closed and the channel will become inactivated. When the channel is inactivated, so no, although the activation gate is open, the sodium is not entering through this voltage gated channel. That means intracellular positivity is gradually reducing. Who will sense it? It will be sensed again by S4. So now the S4 will come down and there will be detachment of alanine and IFM moiety and the channel will be closed. So in this way, it will move into this 4 state. First is closed, then active, then inactivated, then again it will come to the closed state. So this is about how in the 3 state the changes of voltage gated sodium channel will happen. So here we can say the opening of S4 is moving up and the channel is activation gate is opening. So after certain time, the inactivation on the right hand side picture, we can say the inactivation gate is closed, and after that what will happen, again the activation gate will be closed. Now come to the selectivity. So in the previous picture we can see here, other than activation inactivation gate, at the central part of this channel you can see a light blue colored thing is present. So that is the selectivity filter. Why in the sodium channel only the sodium ion can pass, not the potassium ion? Okay, so this is known as the selectivity of the ion channels. If you look into the structure of the ion channel diameter, these ions, they are surrounded by water particles. Okay, you can see the water particle line potassium. It is smaller in diameter, and diameter or the pore size of the potassium challenge is smaller. Okay, whereas in case of, so it is obvious that larger sodium cannot pass through this small pore. This is clear. But in case of sodium ion, you can see that the hydrated sodium ion, it is larger, and the pore diameter is also larger. So it is accommodating sodium. So why can't it accommodate the potassium, which is smaller diameter? The answer remains into this selectivity filter. So when an ion will try to negotiate through the pore, it has to pass through two barriers. One is the outer pore, obviously, and the second at the other pore is called the selectivity filter, which is located midway inside the channel. So when the iron enters the outer pore, which is water, and when it enters into the selectivity filter, so iron has to shed most of its water shell, and because as like us, when we after death we have to leave everything and go away. So in this way, if the iron wants to pass through the selectivity filter, it has to shed most of its water shell. Okay, and how this dehydration will become possible? Because this ion will form a weak electrostatic bond with the polar residues of the amino acid that lines the channel wall. In this picture, we can see these polar amino acid carboxyl groups. What are those amino acids? Glycine, tyrosine, valine has 76 place, threonine 75 place. So they have their carboxyl residues present over here, and the ion has, they are forming a bond with them. Okay, now this, if this bond formation is close, that is, it is closely fit, if the ion is very closely fit with these selectivity filter amino acids, then the energy that is released will be adequate or optimum to cause to stripping off the water shell. Okay, in case of sodium, you can see that here the bond formation is there, but it is not closely fit. It is actually floating loosely within the channel. So the energy that will be formed, that will be released by this bond formation, will be inadequate for stripping this sodium water shells of the sodium ion, and it will not be able to pass through this potassium channel. So similarly, so ion has to it is same for all other ions. They has to pass two gates. One is the outer pore, another is the selectivity filter. In the selectivity filter, the water shell has to be stripped off, and it is only possible if the ion fits closely with the carboxyl residues present of the amino acids lining this inner wall, that is at the selectivity filter. Okay, so this is the reason of the selectivity of these ion channels. Thank you. Uh-huh. And the reference is the Principles of Medical Physiology, Dr. Shobboshachi Sarkar, second edition.